Good afternoon, everyone. My name is Antoine Adamantidis, and I'm a professor of system neuroscience at the University of Bern in Switzerland. And today, I would like to talk about the sleep and the hypothalamus, and we'll focus on work that we've done in my lab and other, essentially in mice, where different techniques have been used to understand how the hypothalamus controls the sleep-wake cycle. So this is a representation of a normal sleep-wake cycle with four in this case, different sleep stage, non-REM sleep, and then REM sleep. What is important to realize here is that the oscillations that are recorded in human ear on the left part of the slide are extremely similar to the one that can be recorded from the mouse brain that are shown on the right part of the slide. And so based on EEG here in blue and EMG in red, one can define the state of wakefulness, non-REM sleep, and REM sleep. And clearly, you can see here spindle in mice that can also be observed in human during stage 2, and this large slow wave here during stage 4 that I've seen here in less than 1 Hertz oscillation in mouse and the delta oscillation. And theta rhythm is quite obvious during REM sleep in mice and similar to human. So this is a short overview of what we know about sleep in the physiology of sleep. We know that it is homeostatic and a circadian regulation of sleep. I'll show you that there is many different heterogeneous cell types that are involved in controlling sleep-wake states, and those cells are broadly distributed across the whole brain. That suggests there might be some redundancy or specialization in neurons controlling those transitions between sleep and wake. There have been a paradigm shift from global to local sleep, where sleep now is considered to be locally regulated. And that has induced a profound change in how we approach the mechanistic role of neural circuit in sleep control. Although we know more and more about different cell types that are either active or silenced during sleep-wake states, there is still no unifying sleep-wake model. So a lot of work is still needed to understand how the brain and the body control sleep-wake states. I put this slide just to remind you that there are currently 2 views in the field that explain how the brain, in this case here estimated to the brain, control the sleep-wake cycle. One is called the circuit view and posit that there are specific circuits in the hypothalamus, but also other parts of the brain that control either wakefulness, non-rapid eye movement sleep, or rapid eye movement sleep, REM sleep. And interestingly here, the hypothalamus is a preeminent structure as it contains neurons that are controlling both wakefulness in the lateral hypothalamus, posterior hypothalamus, but the anterior hypothalamus includes neurons from the BLPO or MNPO that control non-REM sleep. So really there is this switch already in the hypothalamus, but that is also expanded to the whole brain structure that controls sleep-wake cycle through mutual inhibition. Another view is the oscillation view, where sleep is considered as emergent properties of cellular network, where local oscillating network or cellular activity in the brain can generate specific oscillation, sleep oscillation, and that sleep can therefore start locally and reach a global state by expanding and recruiting larger part of the brain. Although those are 2 different models, there are data now suggesting that they might coexist in the brain. And to further illustrate the role of the hypothalamus in sleep-wake cycle, I've represented on this slide the sleep-wake cycle from wake to non-REM and then REM sleep. And I listed many different cells whose activity correlated either with wakefulness in green, non-REM sleep in blue, or REM sleep in dark blue. And interestingly, you can see that there are cells in the lateral hypothalamus, in particular the hypoketonorexin neurons, or the histamine cell in the tuberomammary nucleus in the posterior hypothalamus, that are strongly involved in inducing wakefulness from sleep and probably maintaining wakefulness as well. In contrast, in the anterior hypothalamus, in the ventrolateral preoptic area or the median preoptic area, there are different cell types that either, that most of them are inhibitory and produce GABAergic, but also other peptides, including galanine, CCK, TK1, whose activity is quite high during non-REM sleep, and their activation using optogenetic or pharmacogenetics really increase the occurrence of non-REM sleep, and therefore all these decline in the anterior hypothalamus are thought to promote sleep, non-REM sleep mainly, but in some cases also REM sleep. Interestingly, some of the cells, the MCH cells in the lateral hypothalamus, are known as a strong promoter for REM sleep. So you see that within the lateral hypothalamus or the hypothalamus, there are many different cell types that control sleep-wake states. So somewhat about the hypothalamus. The hypothalamus is a small structure at the bottom of the brain, and it involves in many vital functions, including the regulation of body temperature, heart rate. There are cells that are known to control hunger and thirst, sleep and wakefulness, motivation, but also reproduction, aggressive behaviors, and mood in general. Some of those neurons release peptide or hormones, either in the pituitary gland or in the brain. And most of those seem to act are neuromodulatory system in the brain, in the sense that they are produced by a small number of cells in the lateral hypothalamus. This is the case of, for instance, of the MCH and the hypocretin. And those cells project widely through the brain, where they act on many different systems. So the hypothalamus can be divided in different nuclei. This is what is shown here in human, and in the middle illustration in mice, based on either classical cellular staining, as shown on the left, or by gene expressed by restricted number of cells in the hypothalamus. This is the case here of, for instance, the hypocritin, the MCH, or the POMC, CART, and the MPL, AGRP neurons, and the arcade nuclei that are involved in hunger. Interestingly, those genes define very special cell population. But even within those cell population, there are high diversity of different cell types. And this was shown a few years ago by RNA sequencing. And I'm showing you here the up to 15 different subpopulations that one can isolate from lateral hypothalamus inhibitory into GABAergic neurons in the mouse brain. So really there is a high complexity, not only in the anatomy of the hypothalamic cells, but in the genes that they express and all the other markers that can be found. So how the hypothalamus integrate input and modulate physiological function is represented here. So it receives direct input either from the environment or the ecological niche, and internal states, so to metabolic factor, endocrine, hormone, but also input from brain region involved in emotion, arousal, to neuromodulation. And so I'm showing you here 3 different levels of the hypothalamus from antero posterior axis. And many different cell types, as we will see in the next slide, integrate those responses into either physiological homeostasis or behavioral homeostasis. So what is defined here as physiological homeostasis is thermoregulation, endocrine, metabolism, while behavioral homeostasis is defined by sleep-wake regulation, feeding and drinking based on metabolic need, reproduction, or fight or flight response. An important aspect to emphasize here is that each of those either physiological or behavioral homeostasis is regulated at a very specific time scale, meaning that the neurons that are recruited during those behavior act on the timescale of minutes up to days, as is the case, for instance, for parental behavior or energy store and molecular adaptation. If you're interested in this, please, we just published a review on this. So the general functioning of the hypothalamus, as I just showed you, is that there are cells that are involved either in wave or non-REM or REM sleep. Therefore, there is always this yin and yang type of regulation. So shown here as, for instance, wave-promoting neurons, but also REM sleep-promoting neurons. And of course, those cannot be activated at the same time. So there is probably reciprocal inhibition in between those and or feedback from postsynaptic target back onto the hypothalamus. So this is a functioning scheme that help us to understand how the hypothalamus regulate sleep and wakefulness, but the detail of this connection is far from being understood. So this is the subject of intense animal study. One particular aspect that I wanted to share today is that some of those cells in the hypothalamus that control either thermoregulation, sleep, or wakefulness are located in the anterior hypothalamus, but also in the lateral hypothalamus and the posterior hypothalamus, as I just showed you in previous slide. But interestingly, some recent study found that cells that are involved in the control of sleep might also be involved in the control of thermoregulation, suggesting that single cell within the hypothalamus, this is the case in the anterior hypothalamus, some single cell here might share function. By sharing function, I mean they can modulate either sleep or thermoregulation or both. So that has many consequences conceptually on understanding how the hypothalamus controls sleep-wake cycle, because it might means that sleep, as you know, is always accompanied by decrease in body temperature. So the question remains as whether one is the consequences of the other, or not at all, or whether some of the cells share function while others are more specific to sleep and thermoregulation. So this gives you a state of the research, and there's many perspective and open question to what we know about the hypothalamic cells. Actually, many different cell types, but there are still some question about is there other hypothalamic cells involved in regulating the sleep-wake cycle. I mentioned neurons, but astrocytes are obviously also involved in the hypothalamus. What are their role? How they contribute to the regulation of sleep-wake cycle is still not very clear. I show you that some neurons control both sleep and body temperature. So we still don't know whether those are the same cells, different cells, overlapping cells. This is the case for body temperature, but it's also the case for reward circuit or other physiological function like stress or fight and flight response, reproduction, for which some of those cells might also be involved in sleep, or at least active during sleep, and we still don't know what that means. So eventually, I think, although we focus on the hypothalamus here, it's important to put the hypothalamus back into the brain and see how those cells connect and modulate and communicate with other circuits in the brain that are also involved in the control of sleep-wake states, and eventually also the body control of sleep. This is still an open question, and I hope that in the next year or so we'll have more answer to those. Thank you for your attention.